Hello, welcome to Biology, the scientific study of life. Environmental cleanup. Increasingly, the remarkable ability of certain microorganisms to transform chemicals is being exploited for environmental cleanup. Scientists are now engineering these metabolic capabilities into other microorganisms, which are then used to help treat some environmental problems. For example, many bacteria can extract heavy metals such as copper, lead, and nickel from their environments and incorporate the metal into compounds such as copper sulfate or lead sulfate, which are readily recoverable. Genetically engineered microbes may become important in both mining minerals, especially as ore researchers are depleted, and cleaning up highly toxic mining wastes. Biotechnologists are also trying to engineer microbes that can degrade chlorinated hydrocarbons and other harmful compounds. These microbes could be used in wastewater treatment plants or by manufacturers before the compounds are ever released into the environment. A related research area is the identification and engineering of microbes capable of detoxifying specific toxic waste found in spills and waste dumps. For example, bacterial strains have been developed that can degrade some of the chemicals released during oil spills by moving the genes responsible for these transformations into different organisms. Bioengineers may be able to develop strains that can survive the harsh conditions of environmental disaster and help detoxify the wastes. Agricultural applications. Scientists are working to learn more about the genome of agricultural important plants and animals, and for a number of years they have been using DNA technology in an effort to improve agricultural productivity. Animal husbandry and farm animals. DNA technology is now routinely used to make vaccines and growth hormones for treating farm animals. On a still larger experimental basis, scientists can also introduce a gene from one animal into the genome of another animal, which is then called a transgenic animal. To do this, they first remove egg cells from a female and fertilize them in vitro. Meanwhile, they have cloned the desired gene from another organism. They then inject the cloned DNA directly into the nuclei of the fertilized eggs. Some of the cells integrate the foreign DNA, the transgene, into their genomes and are able to express the foreign gene. The engineered embryos are then surgically implanted in a surrogate mother. If an embryo develops successfully, the result is a transgenic animal containing a gene from a third parent that may even be for another species. The goals of creating a transgenic animal are often the same as the goals of traditional breeding. For instance, to make a sheep with better quality wool, a pig with leaner meat, or a cow that will mature in a shorter time. Scientists might, for example, identify and clone a gene that causes the development of larger muscles. Muscles make up most of the meat we eat in one variety of cattle and transfer it to other cattle or even to sheep. Transgenic animals also have been engineered to be pharmaceutical factories, producers of a large amount of an otherwise rare biological substance for medical use. For example, a transgene for a desired human protein, such as a hormone or blood clotting factor, can be inserted into a genome of a farm mammal in such a way that the transgenic product is secreted in the animal's milk. The product or the protein can then be purified from the milk, usually more easily than from a cell culture. Recently, researchers have engineered transgenic chickens that express large amounts of the transgenic genes product in eggs. Their success suggests that transgenic chickens may emerge as relatively inexpensive pharmaceutical factors in the near future. Human proteins produced by farm animals may differ in some ways from the corresponding natural human proteins. These proteins must be tested very carefully to ensure that they will not cause allergic reactions or other adverse effects in patients receiving them. Also, the health and welfare to farm animals carrying genes from humans and other foreign species are important issues. Problems such as low fertility or increased susceptibility to disease are not uncommon. Genetic engineering in plants, agriculture. Back to this figure, notice that farm animals, these transgenic sheep, carry a gene for a human blood protein which they secrete in their milk. This protein inhibits an enzyme that contributes to lung damage in patients with cystic fibrosis and some other chronic respiratory diseases. Easily purified from the sheep's milk, the protein is currently under evolution as a treatment for cystic fibrosis. Genetic engineering in plants. Agriculture scientists have already endowed a number of crop plants with genes for desirable traits such as delayed ripening and resistance to spoilage and disease. In one striking way, plants are easier to genetically engineer than most animals. For many plant species, a single tissue cell grown in culture can give rise to an adult plant. But those genetic manipulation can be performed on a single cell, and the cell then used to generate an organism with the nutrients. The most commonly used vector for introducing new genes into plant cells is a plasmid cell, called T-plasmid, from the soil bacterium Agrobacterium tumefaciens. This one, tumefaciens. This plasmid integrates a segment of its DNA known as tDNA into the chromosomal DNA of its host plant cells for vector purposes. Researchers work with a version of the plasmid that does not cause disease, as the wild-type version does. Notice this figure outlines one method for using the Ti plasmid to produce transgenic plants. Scientists can introduce recombinant Ti plasmid into plant cells by electroporation. Alternatively, the recombinant plasmid can be put back into Agrobacterium susceptible plants, or plant cells growing in culture are then infected with bacteria that contain the recombinant plasmid. Destructive microbes and insects have reduced the need for chemical insecticides